Cyril.
Yeah. Thanks, Vasant. And hello, everyone. Let me share my screen. So, good morning, good afternoon, and good evening, everyone. And as this title says, we are going to discuss a lot rather than a presentation. And I have been in the research field for a few years and with different groups. So, I like to share what I think or what I get to know that the things that actually makes a difference or the things that are very important for the reviewers or the readers so that your publishing process or your presentation actually has an impact on them. So first I'll start with the publishing part. In the publishing, it's a process that has several steps, and these steps can be utilized to influence the reader. So, what are the steps? Initially, when you collect all the data, you have nice experiments. You collect data, you analyze them, and now you know a story in your mind. And the first step is to put it as a title. So that's the very first thing that you're going to do before start to write your manuscript. Then the next step is to make a small abstract and then figures, and then you're going to decide the journal, because then you have to optimize the figures and the drafting based on the journal requirements. So you are going to draft based on the journal you decided. Then after drafting the final version, you're going to submit it, and the reviewer is going to review it. Then they may have questions, and you are going to reboot those questions in the next step. And all of these steps are very subjective. That means every person have their own opinion on how these things should be. So it's extremely hard to generalize what is correct, what is not correct. But one thing that we have to know is that all these choices are going to influence a reader. And in the publishing process, your first reader is going to be your reviewers. So in the publishing, directing the reviewers to the core points of your paper is the key point. And for that, you have to know how the reviewers get to know your paper or get to know your manuscript. So initially, the reviewers will receive an email from the journal editor with your title and abstract. Then the reviewers decide whether they have interest and time to review that paper or not. So the very first encounter is the title and the abstract of your manuscript. So those are very crucial because your paper or your manuscript is going to be judged from the very beginning. So you have a title, you have an abstract, the reviewers are going to read, and then they are going to decide whether they want to spend more time on reviewing your article or not. And if they accept to review it, then the very next thing they're going to look at is the graphical figure, the figure one in your manuscript, the graphical figure. And then they're going to read or look through all the figures that explains the results of your experiments. And finally, they're going to read the conclusion. And I'm sure, and I have spoken with some reviewers, and I myself review papers. This is the general pattern. They are not going to dwell in the reading of your lengthy introduction or very deep scientific discussion in the very first go. They're going to look at your title, they are going to read your abstract, and the figure one is going to make a huge impact on them because they are trying to visualize what you do based on the image they look at. And then they're going to try to understand your results just by looking at the figures rather than reading all the explanations. Then they are going to read a conclusion. So this is initial 15-20 minutes of their time to get to know what your paper is. So as many people say, the first impression is the best impression. So if you get them in all these steps, then you get them. Otherwise, they are not going to follow your paper very well, or they are not going to follow the paper as you want them. You want your paper to be followed. So the main importance of the title is that it has to show the significance of your research. For example, if we are not going to do the same research again and again, every paper has a difference and significance. So it's very important to show what your paper is about and why it is important. For example, if you do some, if you write a manuscript that is first of its kind, then it's absolutely okay to mention first of its kind in the title. So that's going to give them the importance already. And in the abstract, they are going to know what it is. So it's not the abstract is not going to be based on the results, but it's going to tell the story without strong conclusions. And the graphical figure, yeah, the whole research, the whole manuscript is going to be represented in a graphical way so that the reviewers or the readers can visualize what you do. And of course, they are going to go through all the figures. If you have 10 figures, they're going to look at all the 10, and it should be clear and it should not be tiring. Because if the reader gets tired, then they are going to lose their interest. So it should be clear and less tiring. And of course, the conclusion, now you already explained what in abstract. So you have given what it is about, but after that you explain a lot of results. Now you again going to say what the whole research is about, what the results that you got, and based on the results, you have to give some perspective to the reader. So yes, this is the thing so far, and based on that, so what? What next? You can give some perspective, your own perspective. So that's the conclusion part. That gives a whole story even without reading the 2-3 pages of text. And in the in these steps, most of the reviewers are going to spend their longer time in graphical figures and all your figures that shows your results. So that's going to occupy the reviewers in the reading process. So it's it's good to give some importance in the graphical figure and also the figures that explains your results. So as I said, now your figure is going to narrate your scientific story to the reader. So what are the important things that we can pay attention to? First, the quality, the representation, the colors that you use, the alignment and the space the figures occupy, and of course, in the end, the caption. I would say the caption is less important because the figures should be self-explanatory. And of course, if you cannot explain everything in the figures, you can explain them in the caption. But in the very best, the very best way is to make the figures self-explanatory. So now I'll speak more about the quality and the colors because that's going to catch their eyes and then they are going to look at the fine details of your data. I have an example on the left. You have an image that is saved in the JPEG format from Excel, and I did not change any colors. So this is the very usual outcome if you plot some data in Excel. And the quality is not great. And I did not align the spacings, or I did not even align the axes. And I have another image. It explains the same data set, but with different colors, more sharp colors, not different colors, more sharp colors. And I also have changed the size of the axes to make it look better. And still, the quality is not great. I'll show why. If you zoom that, the first two image, okay, I'll go to the first image. Now you can see the text and the data is not very clear. It's in the JPEG format. And the second image is again in the JPEG format, but with brighter colors. Still the quality is not good. But I have a third format. If you look at these two now, in the third format, you even have your data very clear, even when you zoom it to very high percentages. So Now I'll explain how to do this. So the colors. Most of the scientific figures are based on the complementary colors. So you have the color wheel, and you can use that color wheel to choose the colors. And if you see my previous image, I used orange and blue because they are complementary. They are orange and blue that can catch your eyes very easily. And you can also set a color theme for your whole manuscript. You can make all your figures and your data sets in the same 2 or 3 colors that can make the readers bit that can make the reading bit easy to the readers, because they don't need to switch to different colors for every figure. If you have a pattern, then their mind already knows what the pattern is, so they are not going to struggle with understanding the data sets. And you also have the eyedropper tool and custom color tools, color option in Excel and PowerPoint. Please use them to fine tune your colors, because in Excel, in the default version, you will be having one orange and one blue, but you can of course change them to different different intensity and more brighter or less brighter. So it's good to use the eyedropper tool and the custom color panel in Excel. So this is about colors. If you pay attention to these colors, you're already going to keep the reviewer occupied. And the second thing is quality. So there are types of images, that's vector based images, because most of the image, if you save in JPEG, images are going to be saved based on the pixels. But there are other options where you can save images in the vector based version. So the vector based versions save the images in such a way that even if you zoom them or enlarge them to a great extent, they are not going to lose their quality. So how to do that? In If you want to do save your graph or your figures in Excel or PowerPoint, you can click the graph or the image, or right click the graph or image, and there is an option called Save As. Then in the Save As option, you have the types in which you want to save. Mostly we go for JPEG or TIFF, but if you look at the bottom, there are two versions: scalable vector graphics format and also enhanced Windows meta file. Both of these can have this vector based saving. In the previous slide, the last image that I showed is saved with a scalable vector graphics format. I think the previous versions of Excel and PowerPoint has only Windows enhanced Windows meta file, and these two are pretty much similar in terms of quality. So I with in this presentation, I only want to concentrate on this colors and quality. So now you know you have some options in to make these colors and the quality of your figure look better. And with this, I'll conclude the publishing part. We can, of course, at the end discuss all other parts if we need. Now I'll go to the presentation, scientific presentation. So the very important thing is in the scientific presentation is knowing the audience. So what are the types of audience you're going to have? It's mainly the topic specific audience and general audience, who are very different from this topic specific audience. So the topic specific audience, they already have the background of what you do because they are already in the field. They have fast understanding because they already have the background, and they are more curious about the new results that you have. But you also going to have general audience. They are not specific to the topic. They can still be scientific audience, but then they are not necessarily from the same background or same topic that you are going to present. So you have to cover them as well. So it's going to be difficult for you or for me or for any presenter to comfort both the readers and the general audience. They are probably going to getting to know the topic when you present, so it's just, everything is going to be very new. They are not going to understand everything faster, and also they are not going to be curious about your results. What they want to know is the big picture of what you do and why you do. So if you can explain these things, you already have those audience in line with your presentation. So it's always better to spend the first 2-3 minutes or 5 minutes based on the length of your presentation to cover the general audience. So I'm trying to, now I consider you people as my audience, and I'll try to convey my research topic to you and see if you people can understand it without much difficulty. So if I need to have you with my flow in the presentation, then as a general audience, then the first thing that you are going to look at the slide is the title. So should the title always have the general topics like results and discussion or experimental conditions? No, it's not necessary. It can be a statement. And of course, I have to explain everything using graphical representations because that is easy for a common audience to visualize, probably with simple animations, so they can keep up the flow of my speech or how I explain my research work. And of course, importantly, I have to show why I'm doing and what I'm doing. Now let's go to my research topic, and I'll try to explain you my research topic. And if every one of you understand it, then this discussion has something to learn. I work in a technology called capacitive deionization. It's actually a desalination process using two porous electrodes. So this is how a desalination cell looks like. Not exactly the cell design, but this is the representation. So I have two porous electrodes that is separated by a spacer. And of course, I have current collectors to connect the electrodes to a current source. And I have feed water that contains salt ions, positive and negative ions. And I'm going to apply a voltage difference between these two electrodes. So what happens is that electron runs from one electron to the other. So where the electron goes, that electrode is going to get more negative, and the other electrode is going to get more positive. So what happens? The more positive electrode is going to accept the negative ions from the solution, and the more negative electrode is going to absorb the positive ions from the solution. So the positive electrode absorbs the negative ions, and the negative electrode absorbs the positive ions. And the water that comes out of the cell is desalinated already. So I'm getting fresh water. And these electrodes are not infinite, and it doesn't have infinite storage. So at some point they will be saturated with the ions that are absorbed. So I have to dissolve them. What I do, I apply the negative voltage. So now the electrons runs on the other side. Initially, it was running from the bottom electrode to the top electrode, and now it comes from the top electrode to the bottom electrode. That means the electrode that I made positive and negative is now going to be reversed. So all the ions that are absorbed due to the positive charge or negative charge of the electrode is now going to be dissolved. That means my water, the water that comes out of the cell is going to have huge lot of ions. So I produce brine. And once I dissolve all the ions, I can again repeat this process. So it's a cyclic process of desalinating and produce and brine production steps. How do I follow this process in the lab experiments? So this process, it can be monitored continuously. How? I have salt ions in water, so the water is conductive. And during the desalination process, just by measuring the conductivity, I can know actually the concentration of the salt. For example, let's say I have a solution just with sodium chloride, and every concentration of sodium chloride has its own conductivity. So if I know the conductivity, I can calculate the concentration, and that's what I do in my research. So I ran my desalination cell for some times, and I monitor the concentration, in other words, the conductivity, and that is going to change up and down. How? The dotted line is the concentration of the feed water that I flow through the cell. And when I apply cell voltage, for example, when I apply a positive cell voltage, because in the right y-axis you have cell voltage, and in the left y-axis you have concentration. When I have positive cell voltage, the ions are going to be adsorbed. So the water that comes out has less conductivity, and in the other words, less concentration. That means I'm desalinating the water. And in the next step, I'm going to reverse the cell voltage. So now my cell voltage is going more negative, and I remove all the ions that are adsorbed from in the electrodes. So everything now comes to the water. So my water has more and more uh, concentration. So it's going to be a adsorption step followed by a desorption step. So during the adsorption, I desalinate, and during the desorption, I actually produce brine. And this is what I do in my PhD. It's a simple explanation of my research, and I hope you understand what I do. And if you notice, generally I was trained not to show too many graphs in a slide. That will, if I have 3 or 4 or 5 graphs, then it's going to keep the reviewer, sorry, review, not reviewer, the audience very confused. So it's always better to show 1 or 2 graphs, and it's always good to show them one by one the data sets as I shown now. I initially showed only the concentration and then the cell voltage, and then I also explain how it works. So this is about today's presentation. So I would like to summarize few things. So in publishing, of course, you spend 1 or 2 years in your PhD to collect all those data, but if you don't communicate it well to the reviewers, then you're losing something that you deserve. So it's always good to spend more time on making your figures and choosing a proper title and spending some time in the abstract. So these things are very crucial, but not everyone pays attention to these things. And the second thing about the presentation, of course, nowadays every scientific conference is multidisciplinary. So people from different background comes in, and you have audience from every corner of the world with every different background. So it's always good to have everyone in your presentation. You have to make sure that everyone understands what you do and why you do. And of course, you can be very specific to the topic specific audience in the later part of the presentation when they want to know the results. But in the beginning, it's always good to have both the audience in the flow. So that's it about the presentation. And with this, I'd like to thank Vasant for this opportunity. And of course, I discussed with my colleagues to make this presentation. So thanks for them and my mentors. And if you have any questions, we can of course discuss. And you all might have already published, or you are going to publish. You have different opinions. Please feel free to discuss now, and we can actually make all those key points here and let everyone get benefited from them. Thank you, guys.
Yes.
Any questions?
Thank you, Mr. Searle, for the wonderful presentation. I hope the audience will have a lot of questions to ask. Like the forum is open for discussion. If someone have gotten any question or some discussion or a statement to discuss, you can come up so that it will be beneficial to you as well as for the other audience. Now the forum is open for discussion.
Hello, sir. Your presentation was very nice.
Thanks.
Hello. Hello.
Yeah, I'm hearing. Hello.
Yeah. Okay, sir. I'm started just now. I'm starting my research career. Actually, I have a doubt. When publishing paper, we have related some natural words. If we are using water as a solvent, it will go green chemistry. This much level only I know. But
Mhm.
how I choose Is this paper is publishing in green chemistry or low impact journal? What is that borderline, sir? Actually, this much of work was suitable for that one. I have a doubt if it is possible in published in high level journal or it will go in under level journal only. How I determine that borderline? Any criteria is there for
Um,
that?
yeah, thanks for the questions. I would say the impact factor is not the first choice. You have to choose the journal that is more suitable to your research. And if you have a few journals as an option, which is related to your research, then you can aim for the best journal that you think uh, that will be suitable for your research. And what I can say is that if our results have 50 percentage weightage, then the way that we communicate those results have 50 percentage weightage in the publishing process. So it's very crucial that how you present your results to the reviewers or the readers. So I would say first choose the journal wisely, then make all your efforts in a in the presenting in the way that you present your results, and I think that will definitely make a change. I
Okay,
hope
sir. Thank you.
I give a proper answer to you.
Yes, sir. Thank you.
Nice presentation, Sri.
Thanks.
Can you comment the file size? For example, vector image, if I save, it may be larger size, right?
Yeah.
Can you comment on it?
Um, the thing is, uh, I recently published a paper an year ago, and I used almost all the vector images. But the thing is, the main paper always have less images, or I try to keep the images less in the main text. So my main file never exceeds the requirement of the actual the journal's requirement. My main paper had only 4 images, and all of them are vector-based images, and I didn't have any problem with the size. So I think if you go for if you can make figures uh, that can explain a lot of results instead of so many figures, if you can concise them, you can also concise the size. So I think that should be one of the options, because if you sacrifice your quality or your the quality of the colors or the quality of figures, that's not going to work out well.
Okay. Thank you. Nice
Yeah,
presentation. Okay.
thanks.
Cyril, there is a question in the chat box. Like I think like you'll be able to read it. I'll repeat it for the
Uh,
beneficial of all the audience. It was like very nice presentation, Cyril. And the question is whether the size of the image will be larger when the image was changed to scalar or vector format. I think we have answered it.
yeah,
Yes. So I think the participant is satisfied. So I have got one question like why do we have why I know I can understand how this color scheme is important for the figures, but what is my
mhm,
question is why this color scheme is more important like than the data. Suppose we have got a good data and then
mhm.
the results are very good, but what is the impact of this color scheme then suppose we have got two images where this color scheme is followed and there is another image where the color scheme is not followed. What is the
Mhm.
actual difference in the impact?
You can of course have different figures that cannot be adopted within this color scheme. But what I say is what you represent or what you present, you make it more enjoyable. You can just go with black and white. That's absolutely fine. But differentiate your data sets with different symbols at least. So the whole concept is to make the image easily understandable. So in my opinion, the colors help. And for example, before the digital era, a lot of people were going to print out the manuscript. So at that time, it was good to have only black and white, and they differentiated the data sets with symbols. But now with this digital era, we have added advantage that we can use colors. So why not use them? It doesn't make any image more important, any figure more important or less important. It just makes how important they are. You are going to direct the reviewer to the point that you want them to have a look. So it's just a way of directing someone to where you want their concentration.
Yes, I think like I get the answer. Thank you.
Thanks.
And then there is then one more question, like you, because the most of the stress was on the title. So what is
Mhm.
the so on your perspective, on your based on your knowledge, like how lengthy should the title be? Does the title should convey 100% what the paper needs, or it just has the keywords on it?
I would say it's always good to have the keywords only, because you can make a very clear title with 3 or 4 lines, but it's not necessary. So what a reader wants from the title is why your work is significant, and then they are immediately going to read your abstract. So there you can be very specific, very lengthy, and whatever you wanted to say in the title you can say in the abstract as well. So if you can keep the title shorter, it's better.
Yes, thank you. So I think we'll wait for the last two questions. So then after that we'll close the session.
Yeah.
Yes.
And I'm also open to any other inputs from the audience, because I know there are a lot of experienced audience here. So please, if you have any other opinion, please share here.
Yes, I think like we'll have someone to answer it.
Sir, actually, number of reference. If we one work if we do, we did a number of reference more than 100 or more than 200. This like much is that number of reference means a lot of people's doing this work. So he also repeated that like image is formed by the reviewers, or how that affects the our papers, sir.
When I review or when my mentors review papers, we of course check the references, but we don't check the number of references. We actually don't pay attention to how many references they cited. We just, when we read the text, we just want to confirm the text that are already written. We just want to make sure whether the statement is correct or not. So it's, we are not, or the reviewers are not going to judge the paper based on the amount of references you cited.
Yes, sir. Yes, sir. Okay. Okay, sir. If we do this important reference only if we mentioned it is better, sir. Actually, I think. Am I right, sir?
Yes, you definitely don't need counts. All you need is the proper references, probably the original reference. For example, if you use some method that is already published in 1950, it's better to cite the very first paper, the origin paper. So try to give the correct references rather than giving many references.
Yes, sir. Thank you.
Yeah, thanks.
I think with the connection to the reference, I have got, this question will be beneficial to most of the young researchers who started. So, can you comment on the reference quality? Because some people think that the older the reference, the older the data is, or some people think the the reference should be in the like very recent references, or like what is the kind of reference we one should look for when they are writing up? Does the older reference or the original paper which suited out the topic or the latest research topics which they are looking for? And then secondly,
Uh,
like with this I have one more also. because we have two kinds of papers, like though the impact factor is not the only criteria, but there is a smaller kind of criteria, and some people think like citing a low impact factor is not worth for the citation, and then citing the same kind of one work and the higher impact factor is worth the citation. So can you say like which one to prefer and why do they prefer which one?
Yes, let me answer your first question. Uh, Deciding whether to refer the oldest paper or the newest paper is based on whether that particular statement has changed over the year. For example, because I discussed the same thing with one of my colleagues yesterday. Let's say you cite something about Langmuir isotherm. You have the basic equation, and if you use the same equation without any modification, it's better to cite the first paper, because from there on the same equation comes along. But if someone modified it in the recent years and you use that equation in your research, then it's better to cite the latest one. So deciding whether you have to cite the oldest one or the newest one is based on which is more relevant, which is correct to which supports your statement better. So you have to decide based on these things. And secondly, I think my personal opinion, and also I have a better match with a lot of my colleagues, that the impact factor is definitely not the deciding factor in accepting a paper or reviewing a paper. So you can cite whatever the paper that best suits your statement. And nowadays people are going open access. So a lot of open access channels does not have a very high impact factor. It doesn't mean that they don't have great quality. They have great quality. Just because they're open access, people are still refrained from publishing there. But after 10 years, I think the scenario will be very different from now. So the impact factor is not the deciding factor.
I think I get your explanation. So
Yeah,
if I'm not wrong, I'll explain. Like it is to choose the right reference to justify your statement, not the older one or the impact factor or nothing is important. It is the right
exactly.
paper to cite the right statement.
Exactly.
Yes, I think now the audience will understand that the reference is not going to decide their paper. It is how you present, like because we have got and saying which that like the presentation is also equally important as that of the content. I think today's talk has justified that. However good your content is, when you present it well, it will reach the right audience. With this, I'll take up one last question from any of the audience, and then we'll just close off this talk. I think someone is typing it out. Either it is a question or it is a statement of appreciation. Let's see, or maybe some criticism. Okay, it is a very sensitive and important question. How to identify the fake journals? I haven't. I think it's on you.
uh, I don't have an answer for this, and I don't even know what is fake journal, what is a real journal. But every scientific body recognizes some journals. So it's all, if you have a doubt on some papers, it's always better to go and check if they are recognized by the wide, the famous scientific bodies. For example, if they are recognized by the Academy of Sciences in India, if it is recognized by the scientific groups in Europe, or if it is recognized by scientific groups in USA, just check. It's kind of homework that you have to do to find out whether the journal is actually existing or not, or it is actually a journal or not.
Yes, I think like the answer is right. Like I have not also known any of the fake journals available as of as I know.
Yeah,
Yes. Maybe the authentication of the information is very important. Like it's the person's duty to go search and find out the truth. Yes. With
exactly.
this,
Spend some time.
yes. Thank you. Uh, with this quick note, like the takeaways of today's talk is to have a crisp title and a clean figure, and use color wheels for the better representation. And vector-based images can give you a very good look.
Quality
And
of
then
figure.
yes, the quality of the graph. And then the last point is like keep one graph per slide and not too many datas in one graph. Yes. With this, I
Yeah.
thank Cyril for his wonderful, nice presentation. Thank you so much, Cyril, and I thank all audience for being available and making this presentation interesting. Thank you all once again. Thank you so much, Cyril.
Yeah. Thank you.